So we've talked about passive diffusion. Passive transport, diffusion, fine. It's not always passive. Passive meant we didn't need any energy. Active, as you might guess, means that you need some sort of energy involved. We do have something known as facilitated diffusion. And diffusion, be careful because we always think about that as being passive and no energy. But when we look at facilitated diffusion, there's no direct energy use. But even though it's going to still rely on the concentration gradient, facilitated diffusion is going to need high amounts of energy to actually build proteins. And the reason why is that with facilitated diffusion, we're looking for a way to move larger things into the cell. And when we move larger things into the cell through facilitated diffusion, we have to make the holes in the cells bigger. So we essentially have to make channels or tunnels for things to move through. To actually build these things is going to cost us a lot of energy. The other type of transport that we need to look at that's going to cost us energy directly is something known as active transport. So this is a lot of energy going in. So this is facilitated diffusion. You notice this protein, something known as a transport protein, is going to help and open up a larger space through which things can move. So what we're looking for is we're still moving by diffusion. It's still down a concentration gradient. It's larger molecules that are trying to move through. It's going to cost us because of building the transport protein. We actually have specific proteins to water, and they're known as aquaporins. So this actually allows the water to move into a cell more quickly. And actually, this was the 2003 Nobel Prize, was the discovery of these aquaporins. And so looking at how these protein channels are very specific to the diffusion of water. So it's the facilitated diffusion of water. So the difference with active transport is that the cell is going to use a ton of energy for this entire process, and it's going to move things against its concentration gradient. This is different. Everything else we've looked at moves things down a concentration gradient. This is going to move things up a concentration gradient. So in this case, looking at a large number, in this case 4 squares outside, 2 inside, but the cell wants to move those out. So it's going to do that, but it's going to cost it energy every time it does. So now we end up with 6 squares outside, which is what the cell wants, but it costs us energy to do it. And every time we do this process, it's going to cost more and more energy. So with active transport, you are looking to use ATP to move things up a concentration gradient. We'll look at these more with specific examples. So how do we actually get things to move in and out of the cell? There are 2 basic processes. Exocytosis. Exo is out. Cyto is cell. So moving things out of the cell. Endocytosis. Endo is in. Cyto is still cell. So we're moving things into the cell. And now we're looking at not diffusing small particles. We're looking at moving really big things. So one type of endocytosis is known as phagocytosis. So this is essentially cell eating. So this is bringing in large particles by engulfing them and literally surrounding and eating these large particles. Penocytosis is cell drinking. So this is actually bringing in or engulfing large amounts of water. This happens a lot when you're dehydrated or when your cell is trying to get at a nutrient that is dissolved in the fluids outside of your cell. And the third one is something known as receptor-mediated endocytosis. So this is using specific protein receptors on the surface of the cell to move things in. So this is phagocytosis, so literally cell eating. See here a food vacuole that the cell is surrounding and will eventually eat. Penocytosis works the same way. You see here, this must be a very thirsty cell. This is the plasma membrane folding in and bringing in very large amounts. See all these fluids that it's trying to bring into the cell very, very quickly. Now, like I said, it can also bring in dissolved particles. So like here, maybe these are vitamins or monomers that the cell needs, and it knows that they're out there and it'll bring them in large amounts. Receptor-mediated endocytosis is a very specific process. So in this case, looking for specific receptors, the Y is a receptor on the surface of the cell here. When the substrate fits into the receptor, this receptor is going to target the cell or signal the cell to make a little vesicle and bring in large amounts of these. And you see this here on the right side as well. So just 3 different types of endocytosis. Exocytosis we have talked about before, with transport vesicles and vacuoles leaving the cell, moving proteins out, moving waste particles out, all of these kinds of things. It's essentially endocytosis in reverse, but with exocytosis, we're looking at potentially sending out things that are going to be used in other cells and just simply sending out waste.